the structure of RNA. RNA was discovered by Friedrich Miescher in the year 1868. It was also called nuclein when it was first exposed. As we discussed earlier, the structure of RNA is somewhat similar to DNA by the sugar component of RNA. Ribose has a hydroxyl group at the 2' position. RNA does not have the pyrimidine equality that is found in DNA. The presence of this functional group causes the helix to adopt the A form geometry rather than B form, most commonly observed in DNA. Adenine, guanine, cytosine and uracil are the common nitrogenous bases of RNA. Thus, the pyrimidine uracil substitutes thymine of DNA. In regions where purine-pyrimidine pairing takes place, adenine pairs with uracil and guanine with cytosine. In addition to these 4 bases, RNA also has some unusual bases. Most cellular RNAs are single-stranded, even though some viruses have double-stranded RNA. In folded regions, they are formed by pairing of complementary bases by hydrogen bonds. This is the figure showing the structure of RNA. This complex structure will help to store information and catalyze its transmission. Stem loop-like structure, please see the figure on screen, is created when 2 complementary sequences within a single strand come together to form double helical structures. That is, hairpins, as shown in the figure, are formed by pairing of bases within 5 to 10 nucleotides of each other, and stem loops by pairing of bases that are separated by greater than 10 to several nucleotides. These structures may form complicated tertiary structures like pseudoknot. Pseudoknot as shown in the figure. Pseudonodes are formed by interaction of secondary loops through base pairing between complementary bases. tRNA molecules and rRNA molecules also have well-defined 3-dimensional structures. Secondary and 3-dimensional structures also have been recognized in mRNA, particularly near the ends of molecules. Stem loop. It's a lollipop-shaped structure formed when a single-stranded nucleic acid molecule loops back on itself to form a complementary double helix, which forms a stem topped by a loop. Hairpins are a common type of secondary structure in RNA molecules, looks like a loop or a U shape. Pseudoknot. A pseudoknot is minimally composed of 2 helical segments connected by single-stranded regions or loops.